Welcome to Sciencera. In today's lesson, we're going to discuss cells and tissue. Starting with the overview of cellular basis of life, we're going to then discuss the anatomy of generalized cells. So, the learning objective for today's lesson is concept of cellular theory, elements that make up bulk of living matter, definition of generalized cells, three major component or region of cell which include nucleus, cytoplasm and plasma membrane, and then we're going to list the structure of nucleus and explain the function of chromatin and nucleoli. Let's begin. Overview of cellular basis of life. Robert Hooke used a primitive microscope to examine some plant tissue cork in the late 1600s. He noticed certain cube-shaped structures that are reminded him of monasteries' length row of monks' chambers. So he termed them cells. Whatever its form, however it behaves, the cell contains all parts necessary to survive in the changing world. It follows then the loss of cell homeostasis underlies virtually every disease. Chemical analysis of cells shows that they are made up primarily of same four elements: carbon, oxygen, hydrogen and nitrogen, plus much smaller amount of several other elements. Cell theory in 1800s were based on four concepts. Number one, a cell is a basic structural and functional unit of living organism. So when you define cell properties, you are in fact defining the properties of life. Number two, the activity of an organism depend on collective activities of its cells. Number three, according to the principle of complementarity, the activities of cell are dictated by their structure, anatomy, which determines the function, physiology of cell. Continuity of life has a cellular basis. So these were the four concepts on which the cellular theory in 1800s were based on. Next is the anatomy of generalized cell. In general, all cells have three main regions or part: a nucleus, plasma membrane, and cytoplasm. Nucleus is usually located near the center of the cell. It is surrounded by the semi-fluid cytoplasm, which is in turn enclosed by the plasma membrane, which form the outer boundary of the cell. So, three main structures or the region of the cell are nucleus, cytoplasm, and plasma membrane. Starting with the nucleus. Nucleus is also known as the control center of the cell. They are often oval in shape, contain DNA which carry genes, instructions for building proteins. DNA is also absolutely necessary for the cell reproduction. A cell that has lost or rejected its nucleus for whatever reason is destined to self-destruct. Nucleus has three recognizable region or structure: nuclear envelope, nucleolus and chromatin. We're going to look at each of these in detail. Starting with the nuclear envelope. It is a double membrane barrier that separates the nucleus from the rest of the body. A fluid-filled gap exists between two membranes. Two layers of the nuclear envelope fuse at the different places, resulting in nuclear pores. Nucleoplasm, a jelly-like fluid in which other nuclear constituents are suspended, is encased by the nuclear membrane. Next is nucleus. Nucleus contain one or more nucleoli, which are tiny, dark, staining, essentially spherical structures. Nucleoli are the plural for the nucleolus, are the places where ribosomes are assembled in cell. Most ribosomes eventually move into cytoplasm, where they function as protein production sites. Next is chromatin. When a cell is not dividing, its DNA is twisted around protein called histones to form chromatin. a loose network of beads on a string that is strewn around the nucleus. When a cell divides into two daughter cells, the chromatin thread coil and condense to create dense rod-like bodies known as chromosomes. Chromo arises from the word color, mean color, and soma mean body. So colored bodies are the chromosomes. Much like when a stretch spring relaxes, chromosomes get shorter. Chromatin gets shorter and thicker. Three parts of nucleus are nuclear envelope, nucleus, and chromatin. Next is plasma membrane. The flexible plasma membrane is fragile, transparent barrier that contains cell content and separate them from surrounding environment. Fluid mosaic model. Plasma membrane is made up of two phospholipid fat layers that are organized tail to tail with cholesterols and floating proteins strewn about. Sugar groups can be linked to certain phospholipids generating the glycolipids. They have hydrophilic and hydrophobic ends. Hydrophilic polar heads of the lollipop-shaped phospholipid molecules are attracted to water, which is the principal component of both intracellular and extracellular fluid, and hence lay on the both inner and outer side of the membrane. Their nonpolar fatty acid tails, which are hydrophobic, avoids water and line up in the membrane's core, which mean interior. When biological membrane are damaged, their self-orienting property allows them to quickly reseal themselves. The cholesterol serve to keep the membrane flexible while also stabilizing it. Proteins dispersed throughout the lipid bilayer are responsible for the most of the membrane's specific function. Enzyme are type of proteins. Many of the proteins projecting from cell surface are hormones or other chemical messenger receptors, or they serve as the binding site for attaching the cell to the fibers or other structures inside and outside the cell. Most membrane proteins are engaged in the transport. Cell membrane junctions. Cells are bound together in three ways. Glycoprotein in the glycolex act as an adhesive or cellular glue. Waxy contour of the membranes of adjacent cells fit together in a tongue and groove fashion, or special cell membrane junctions are formed. These junctions vary structurally depending on their role. So three types of junctions are there: tight junction, desmosomes, and gap junction. Tight junctions are impermeable. Desmosomes are the anchoring junction, and gap junctions are used for communication. Starting with the tight junction. Tight junction are impermeable junctions that encircle the cell and binds them together into leak-proof sheets. In tight junction, adjacent plasma membranes fuse together tightly like a zipper and prevent substances from passing through the extracellular spaces between the cells. In small intestine, for example, these junctions prevent digestive enzymes from seeping into the bloodstreams. Desmosomes are anchoring junctions scattered like rivets along the side of adjacent cell. They prevent cells subjected to mechanical stress from being pulled apart. Structurally, these junctions are button-like thickenings of adjacent plasma membranes that are connected by the fine protein filaments. Thicker protein filament extend from the plaque inside the cells to the plaque on the cell's opposite sides, thus forming an internal system of strong guy wire. The last one is gap junction or communicating junction. Function mainly to allow communication. These junctions are commonly found in heart and between embryonic cells. In a gap junction, neighboring cells are connected by hollow cylindrical composed of proteins that span the entire width of abutting membranes. Chemical molecules such as nutrient or ions can pass directly through the water-filled connexons channel from one cell to another. So three types of membrane junctions are tight junction, desmosomes and gap junctions. The cytoplasm. Cytoplasm is cellular material outside the nucleus and inside the plasma membrane. It is the site of most cellular activities. Three major components of the cytoplasm are the cytosol, inclusions, and organelles. Difference between cytosol and cytoplasm is that cytosol is a fluid present in the cell membrane and cytoplasm is cell component present inside the cell membranes. Cytosol. The cytosol is semi-transparent fluid in which the other constituents are suspended. Nutrients and a variety of other solutes are dissolved in the cytosol, which is mostly water. Chemical components that may or may not be present depending on the cell types are known as inclusion. Most inclusions are nutrients stored in cytosol or cell products floating in the cytosol. Inclusions can be considered as the cellular penetree where goods are stored until they are needed. For example, lipid droplets are plentiful in fat cells. Glycogen granules are abundant in liver and muscle cells. Pigments such as melanin are abundant in skin and hair cells. So these are all the penetraries where goods are stored in the human body. Cytoplasm organelles. Metabolic machinery of cell is housed in organelles, which are specialized cellular compartments. Organelles are specialized to provide specific duties for the cell, like how organs perform specialized function for the entire body. Some people make proteins, while other packages them, and so on. Different parts of the Many organelles are bounded by a membrane similar to the plasma membrane. These membrane boundaries allow organelles to maintain an internal environment quite different from that of the surrounding cytosol. This compartmentalization is crucial to their ability to perform their specialized function of cell. For example, mitochondria. Let's look at mitochondria now. Mitochondria are lozenges-like or sausage-shaped organelles. The mitochondrial wall is made up of two membranes that are arranged side by side like two plasma membranes. The outside membrane is smooth and featureless, but inner membrane feature cristae which are shelf-like protrusions. The reaction in which oxygen is used to break down meal are carried out by enzymes dissolved in the fluid within the mitochondria as well as the enzymes that are part of the cristae membranes. Energy is released as foods are broken down. Most of this energy is lost as heat, but some is retained and used to create ATP molecules. All cellular labor is powered by ATP and every living cell requires a steady supply of ATP. ATP stands for adenosine triphosphate to carry out its many functions. The mitochondria are the powerhouse of the cells since they supply most of the ATP. Metabolically active cells such as liver and muscle cells requires a lot of ATP and have hundreds of mitochondria that pinch in half to duplicate. Cells that are relatively passive on the other hand have fewer ATP as compared to the active cells. Ribosomes. Ribosomes are tiny bilobed dark bodies made of proteins and one variety of RNA called ribosomal RNA are also present. Ribosomes are the actual site of protein synthesis in the cell. Ribosomes that float freely in cytoplasm manufacture the protein that function inside the cells while others attach to membranes such as the rough ER, which produces proteins that function outside the cell. Next is endoplasmic reticulum. The endoplasmic reticulum is a network of fluid-filled tubes that coil and twist across the cytoplasm. It runs parallel to the nuclear envelope and make up around half of the cell's membrane. It acts as microcirculatory system for the cell. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and the smooth endoplasmic reticulum. Ribosomes are found in the rough endoplasmic reticulum. Proteins generated on its ribosome travel into the rough ER tunnels where they fold into their functional 3D structures. Because almost all building components of cellular membranes are formed either in or on it, these proteins are subsequently delivered to different parts of the cell in transport vesicles, which are little membrane sacs that transport things around the cell. Rough ER is seen in abundance in the cell that produce and export proteins. Then we have smooth endoplasmic reticulum, which is involved in lipid metabolism, cholesterol and fat synthesis and breakdown, as well as drug and pesticides detoxification. It's hardly surprising then that smooth ER is abundant in liver cells. Body cells that create steroid based hormones such as testosterone producing cell in male testes are also affected by smooth endoplasmic reticulum. Synthesis and export of proteins by rough ER. So there are four steps involved in the synthesis and export of proteins by the rough endoplasmic reticulum. Step number one is, as the protein is synthesized on the ribosome, it migrates into the rough ER tunnel system. In the tunnel, the protein folds into its functional shape. Short sugar chains may be attached to the protein, form a glycoprotein. The protein is packaged in a tiny membranous sac called a transport vesicle, and then the transport vesicle buds from the rough ER and travels to the Golgi apparatus for the further processing. Golgi apparatus. The Golgi apparatus is characterized by a stack of flattened membrane sacs containing some of the small vesicles. It is situated near the ER endoplasmic reticulum and serve as the primary traffic controller for the cellular proteins. Its main job is to change, package and ship proteins given to it through transport vesicle from the rough ER in specific ways depending on where they are going. Initially, all proteins leave the Golgi apparatus, collect in Golgi vesicles, which are the little. As protein tagged for export accumulates in Golgi apparatus, the sac swells. Then their swollen end filled with protein pinch off from the secretory vesicle which travel to the plasma membrane. When the vesicle reach the plasma membrane, they fuse with it. The membrane ruptures and the content of the sac are ejected to the outside of the cell, which is shown through the pathway 1 on the screen. Mucus is packaged this way, as are digestive enzymes made by the pancreatic cell. In addition to its packaging for release, the release functions, the Golgi apparatus pinch off sacs containing the protein and phospholipid destined for a home in the plasma membrane, which is shown by the pathway 2 or the other cellular membranes. It also packages hydrolytic enzyme into membrane bound organelles called lysosomes that remain in the cell, shown by the pathway 3. So in pathway 1, Golgi vesicle containing protein to be secreted becomes a secretory vesicle. Secretion takes place by exocytosis. In pathway 2, Golgi vesicles containing membrane component fuses with the plasma membrane and is incorporated into it. And in pathway 3, the Golgi vesicle containing digestive enzymes become a lysosome. Lysosome fuses with ingested substances. Lysosomes. Lysosomes are membrane bags that contain potent digestive enzymes and can be in various sizes. Lysosomes serve as cellular stomachs because lysosome enzymes may degrade worn out or non-usable cell structure as well as the most foreign substances that enter the cells. Lysosomes are common in white blood cells called phagocytes, which are responsible for removing germs and the cell debris. The enzymes they contain are synthesized by ribosomes on the rough ER and packaged by the Golgi apparatus as previously stated. Peroxisomes. Peroxisomes are membranous sac containing potent oxidase enzyme that detoxify a variety of toxic or dangerous components or compounds including alcohol and formaldehyde using molecular oxygen O2. Their primary role, however, is to disarm hazardous free radicals. Free radicals are highly reactive molecules with unpaired electrons that can harm proteins and nucleic acid structures. Free radicals are natural byproduct of cellular metabolism, but if allowed to build up, they can cause cell death. The function of peroxisomes mean peroxide bodies is to convert free radical to hydrogen peroxide, which the formula for hydrogen peroxide is H2O2. Excess hydrogen peroxide is subsequently converted to water by the enzyme catalase. Peroxisomes are abundant in liver and kidney cells, which plays an important role in detoxification. Next is cytoskeleton, an elaborate network of protein structure extends throughout the cytoplasm. This network cytoskeleton acts as cells, bones and muscles by furnishing an internal framework that determines cell shape, support other organelles and provide the machinery for intracellular transport and various types of cellular movements. From its smallest to its largest element, the cytoskeleton is made up of microfilament, intermediate filaments and microtubules. Although there is some overlap in roles, generally speaking, microfilament uh, uh, actin and myosins are most involved in cell motility and in producing changes in cell shape. The strong stable rope-like intermediate filament are made up of fibrous subunit. They help form desmosomes and provide internal guy wires to resist pulling forces on the cell. The tube-like microtubules are made up of repeating subunits of protein tubulin. They determines the overall shape of a cell and the distribution of organelles. They are very important during the cell division. Thank you for watching. This was all about the overview of cellular basis of life and anatomy of generalized cell. In next lesson, we're going to discuss cell diversity and cellular physiology. Thank you.